clubs in the zone raised food to feed 300,000 people so far this year. Fantastic. Well done. And Barry has indicated we're going to carry that on with a district-wide food collection campaign this coming year. So that's a great thing. Also then follow up, Rich already talked about it. We're going to continue to send out emails and ask you to tell us if you're going to be here so that we can be well prepared for each of our meetings. So with that, now it's time to go on to our announcements. And today we have a special visitor with the Naus family. Tara and Joe, can you unmute and tell us how Carter is doing? You might remember a year ago when Carter was born, he spent the first 125 days of his life in the hospital, and our whole club was praying for him. So this is a human success story. Tara, over to you.
Hey guys, we're calling in from Sarasota. We're on our little family vacation. Today is actually a year to date when Carter went in for his open heart surgery. About this time last year, he was on the heart and bypass machine for about 7.5 hours total. So we're just did a little family getaway to celebrate, you know, Carter being here.
Carter, yay! Thanks for praying for us. Thank you, everyone.
Fantastic. Thank you for being on, and thank you for sharing the story of Carter with us and letting us see that he's a normal 1-year-old at this point. We love seeing him grow and thrive. Thank you. All right, our next announcement is a special announcement. Today, we have a member who is celebrating his 82nd birthday. Happy birthday, Ben. We're glad you're with us. Let's give Ben Zimmerman a great round of applause. You might not know that Ben is at home recovering from surgery and from a broken hip, but that hasn't stopped him from joining us on Zoom. And Ben has been a member of this club for over 45 years. Well done, Ben. Happy birthday. Okay, and now we have a classification talk. Ricky Vescovasi. Ricky?
I'm right here. Thank you.
There you go.
Appreciate it. Good afternoon, everybody. My name is Ricky Vescovacci, and I've got a couple of things to share. Why did you choose the Rotary? Well, there are 5 reasons. Network, fellowship, mentor, culture, which is important, and then community engagement. Those are the 5 critical areas that I thought would be important to me as part of choosing the Rotary. Why did you choose your particular business profession? I'm a sales and marketing professional, and I enjoy working with people and also solving business problems. And I really enjoy that. And making money while I'm doing those things is important. And then number 3, part of your job that you find rewarding. Really, the rewarding part of it is, again, building long-term relationships with customers and business leaders and solving problems, as well as financially being rewarded. And then the challenges that I think everyone finds is really building trust with your business owners to make sure that they understand that you can help them with your knowledge and with your skills. And then number 4, the ethical issues that you faced at work, or the 4 waste test and Rotary code of conduct. And I would say, you know, ethical issues are really key, right? And the 4 ways test for Rotarians really help. And how does that help? Well, it leverages the relationships long-term. You build mentorship with your colleagues and your peers, and that's really critical, I think, for all of us while we continue to nurture our careers and our family members, right? And we have succession plans with our friends and family. My background, just to give you a little bit of background, I worked for a couple of Fortune 500 companies. I worked for IBM for about 4 years while I was in college. I worked with Xerox for 23 years, and then I'm currently with a consulting firm called Keypoint Intelligence, where we do e-commerce and data analytics. So that's a little bit about myself and my family. We live in Windermere. I've got 2 beautiful daughters and my wife, Luneria, which works with Calvin Gardner. And we've been here for 23 years, and we love being a part of this. So thank you for having me today. Appreciate it.
Thank you, Ricky. We are always pleased to welcome new members and to hear a little bit about them and get to know them. So if you haven't had a chance to get to know Ricky, please introduce yourself and make a new friend. So with that now, it's time for Happy Dollars, and our Happy Dollars collector today is Calvin Gardner. Calvin?
Thank you, Richard. Appreciate it. I'm really happy to actually be doing Happy Dollars today. Richard actually gave a little history on the history of this building with our club, with Harry P. Lou. I'm going to take 30 seconds and I'm going to give you a little history on Ace Cafe. So a lot of you seen that I've been riding my motorcycle to Rotary because I'm pretty cool. And because Ace Cafe has a long history with motorcycles. So most of you don't know it, but Ace Cafe, the original one's in London. This is only the second one ever built. The first one was built in 1938. It was just a diner originally, and then it became a gas station, then it became a store, but it was just like this big conglomerate gas station, kind of like the thing they're building over there in Ormond Beach. But it unfortunately got bombed out during World War II, got pretty damaged. They kind of limped along, but they rebuilt it in 1949. So coming into the 50s, what was hot? Rock and roll. You know who loved rock and roll? Teenagers. You know what teenagers love? Motorcycles and fast things. So they started riding their motorcycles to the cafes because they couldn't listen to rock and roll on the radio stations. They had to go to any place with a jukebox. Ace Cafe had it. Ace Cafe became this huge thing for cafe racers, which is what you see as the Bonnevilles, which is the bike that I ride. And they got their name because they would come, and the big thing was to put a quarter in the jukebox, or probably 10 cents back then, or a nickel. And you had to get on your bike and race to a certain point and get back before the song ended. So that was the Race Cafe. And then they actually called them the Ton Up Boys. And they weren't a great lot. They were just bad teenagers, truthfully. But they had a lot of fun. But this Ton Up Boys was built based on this Ton Up, which was the goal was to try to hit 100 miles an hour while you were riding on these races. So it became a big deal in the 50s. They had thousands of people come through there. It was kind of a rough crowd, a lot of leather. But and that's the history of Ace Cafe, and they actually brought it back in the 90s. A lot of rock and roll people come and play the one in London. They built this here, and COVID hit, but it's coming back strong. So anyways, thought I'd give you a little history. I'll let you put your Happy Dollars in here. My helmet lights 5s, 10s, 20s, and 100s. So we'll start over here. Thank you, Art. Tom?
Finally, the University of Tampa put an article out last month, and Tommy, this is why I always talk about the Bucs when you're talking about a college team. The Buccaneers are and have been for the last 45 years. They are the official school team of the University of Tampa, because they took our Spartans and our stadium and our facilities, and legally we were the hometown team. So the University of Tampa's whole crowd went over, and they're the ones that were the hosts. We don't have
$50? Anybody? Danny's always good for a 5, or there we go.
I've got 5 Happy Dollars. These 4 guys surrounding me at the table are all part of the board of directors of 1 Million Cups. Ellen Schaefer always also sits on our board. Please get to know these guys. They're potential Rotarians and all wonderful people. And the other one is, Ken was telling me that apparently, is it Crystal? what's the name of the chain? White Castle is building in Orlando, and they just gave some ridiculous number of free tickets to the Salvation Army. Something 10,000.
They open May 3rd.
May 3rd for White Castle. We're all looking forward to it. Thank you, Bill. Thank you. Brian?
Yes, everybody, my name is Bryant, and I have $3 happy dollars for my 3 guests, Monica, Gary, and Danielle, who came to join us today for today's speaker. So I just want to warmly welcome to the Rotary Club of Orlando.
Perfect, thank you. And we got a couple back here.
Hi, everyone. I have $5 happy dollars because this is my first in-person Rotary meeting, and I'm all in.
Thank you very much.
I've been Tamara, so she's got my happy dollars for you guys. But I'm happy to be here with all of you guys. Like I mentioned earlier, I'm from the South Seminole, so it's nice to see the traditions of Rotary going on here. This is what they do at my club and network with like-minded people. My happy dollar is my second happy dollar, because I gave 2, is also that I just started a new job, which is nice to be working during this pandemic that we're still in. So I work for Kyocera. If any of you guys need office printers or office technology, hit me up.
Thank you. Welcome. Our New Zealander.
Hi, thank you for having me here today. It's wonderful to be here. As you all know, I'm from New Zealand, not originally. I'm moving to Orlando. I fell in love with Orlando. And although people are different all around the world, one thing I found the same, that's Rotarians. Great people. Thank you.
Great. Thank you very much. That's gonna do it.
I have $5 happy dollars. For those of you that are on Zoom, this is Rich Ungaro. I just want you to know how much I love and miss David Joyce. So, David, please get well.
Thank you. Ditto.
Thank you, Kelvin. I have 20 happy dollars for our many guests today, and 5 of those because I was able to spend the week in Tampa with my 5 grandchildren, and that's always a great time. So thanks again, Kelvin, for a great job on happy dollars. And now we go on to introducing our speaker with the chair of the Speaker Forum, Bill Pieper.
Thank you, Dick. Good afternoon, everyone. It's a pleasure to welcome our speaker, Dr. Eric Hoffman. He's joining us online. He is a native of Nebraska. He earned his bachelor's in, get this, ecology, behavior, and evolution at the University of California, San Diego, a master's degree from the University of Illinois, a PhD in zoology from Oregon State. He went on to do research and teach University of Central Florida. Please welcome our speaker, Dr. Eric Hoffman.
Let's see. Hello, everybody. Can you hear me?
Yes, we can.
Let me get this loaded here. All right. Are you all looking at this screen with my mouse moving around at my presentation?
We're seeing the preview screen as well. So
You're seeing both of them.
yeah, change it back to where we were this morning or yesterday.
Yeah. Now it's. Oh, now I'm going to mess up.
There you go. Just switch there. Perfect. Click it once. There you go.
Okay. Is that good? All right. Fantastic. All right. Well, thank you very much for hosting me today. Today, I'm going to be talking to you about genetics and the conservation of Florida's wildlife. Just as a brief, a little bit more, I've been here in Orlando for 15 years now at the University of Central Florida. I am a population geneticist, which means that I use genetic variation present in natural populations to tell us a little bit about the natural history and conservation of those species. For my talk today, I'm going to first, I'm going to start talking a little bit about wildlife and wildlife conservation. I'm going to give a brief introduction of how we can use genetics and to address some of these questions. And then I'm going to talk about a case study that I've done looking at the striped newt, these guys down here. And then I'll tie it all up with some conclusions at the end. So let me begin. When many of us think of wildlife, we think of something like a lion, like this, not native here in Florida. Or maybe you think of wildlife being a deer. However, wildlife includes all wild animals collectively that are native typically to a particular region. And so this includes things like squirrels or aquatic organisms like manatees and a wide variety of different taxa. Here I show you 4 American alligators, the southern leopard frog, the Florida scrub jay, and the striped newt. These are all different organisms that actually I've done research on in my lab here at UCF. And in fact, the striped newt is what we're going to be talking about a little bit later. And you might be wondering, well, why do we need to conserve wildlife? And the answer to that is over the past 50 years, I would say that there's been a large amount of habitat loss of natural lands, pollution that has inundated, again, natural lands, and overexploitation of natural resources, and then the introductions of non-native species. And all of these factors have worked together to cause a loss of wildlife, really all over the globe. If we want to tie in genetics now in terms of how genetics can be used, the field used for conservation, the field of conservation genetics itself is a relatively new field. Typically, I'm talking to undergraduates, and when I talk about 1974 as sort of the beginning of the field of conservation genetics, they think of that as being like hundreds of years in the past. I'm assuming that with this crowd that maybe many of us even remember 1974 and don't think of it as being that too long ago. But the field of conservation genetics uses genetic theory and techniques to be able to reduce the risk of extinction of threatened species. And it really did get its beginning in 1974 when a guy named Otto Frankel published a paper, and that paper was titled Genetic Conservation or Evolutionary Responsibility. And in this paper, what Frankel said, and it was really, again, this is sort of the first time that this had been put together, was because we cannot anticipate the future world in which humans will live, it is our responsibility to keep evolutionary options open for future humans. And what that means is that we need to be able to conserve the wildlife that's going to be available to be able to keep the world a better place for humans and the other animals that live here. Since '74, conservation genetics has grown into a relatively large field, largely motivated by the need to reduce current rates of extinction and preserve biodiversity. And what I have here are just a number of different textbooks that are all focused on different aspects or maybe specific aspects of conservation and genetics. To make sure that you're all sort of caught up on your basic genetics, it's important to understand and remember, I'm going to be using words like genes and alleles and loci. And so now I get to give you all a brief genetic reminder. Genes genes are parts of DNA in our genetic code that influence characteristics, typically such as hair color or height, can also impact various aspects of behavior. And when we have different forms of a gene, then these different forms of a gene are called alleles. In natural populations, there's lots of allelic variation. So we can look at, say, maybe a gene for hair color, and we can look into the crowd, and you can see that lots of people have different colored hair, and that different hair color is going to be influenced by genes, by different alleles of the hair color gene. And so here in my cartoon down here, we have a locus which is a sort of another term for a gene. In this case, it's the locus for the flower color gene. And we have one allele for purple flowers and another allele for white flowers. And so this just would show you that these different alleles occur at the same spot on the chromosome, but there's just different variations of them. And it's that variation that actually becomes very important if organisms are going to be able to survive and evolve in the natural world in which they live. And so that genetic, the number of different alleles in the natural population can be characterized as genetic diversity in general. But it's important to understand that genetic diversity can occur at different levels. And so we're going to talk about how this can occur across 3 different levels. We're first going to talk about genetic diversity among species. And so here what you have is what's referred to as a phylogeny or a phylogenetic tree. And that's this drawing and how this drawing links together the relationship of these different mammals. And so you can see here that the mouse and the rat a sort of group form a fork together that is that's relatively close together. Similarly, the horse and the dog appear relatively close together. And by being relatively close together, it's telling us that they're genetically more similar to each other, and that organisms that are further apart, say the possum from the rat or the platypus from the horse, that the genetic distance between them just goes to show that there's different levels of diversity among each of these different species. But in addition to having genetic diversity that can occur just among different species, we can get genetic diversity that can occur among populations within a species. And so here what we're looking at is, again, a phylogenetic tree. It's much smaller this time, and certainly I don't expect you to be able to read all of the different words that follow this tree, but you should be able to follow the general pattern. Here we're looking at genetic variation that exists within a species of chipmunk, and this is out in Canada and Washington and Oregon and California. And what you can see is that there are different groupings. So here, all of these that are yellow form a single genetic group, whereas maybe all of these that are red form a different genetic group. And that there's basically what you see is that there's regional variations among populations within this single species. And so this is due to just genetic population or genetic diversity arising within different populations. And then the last level of diversity that we want to look at is diversity that can occur within a population. And so even within a population, we can see lots of genetic diversity, lots of genetic variation that is present. And so, not just within Homo sapiens, but in maybe a more natural example, here's a population of snails, Sepia moralis. These snails are a classic study because it turns out that the genetic basis for the banding patterns, the genetic basis for the colors have all been worked out. And so we have a good understanding of what causes this variation. But within these populations, we see lots of variation that is present. And then the other bit of genetic background that you need that I would like to be able to teach you has to deal with how these different alleles change between populations or over time. And so here what we're looking at is changes. What are the factors that cause the changes in allele frequencies within and then between populations as well? And so these change in allele frequencies are typically influenced by four main processes. One of these is mutation. And so a genetic mutation causes a single base pair to be changed out from one base of DNA to a different base. It causes, an actual physical change in the DNA of an organism. And in doing so, it causes, the proteins to be changed, and this causes the creation of different alleles within a population. Another process that causes allele frequencies to change within populations is natural selection. And so here I have a cartoon that shows that these birds have a preference for eating the green beetles. And what we can see is that in a population that maybe originally had 3 orange beetles and 13 green beetles, that generations later, you can see there's now 6 orange beetles and 10 green beetles in this population, right? Natural selection is referring here to the differential survival. And in this case, differential survival of these beetles is based on the different coloration of those beetles. The next one, the third process we'll talk about is called genetic drift. And genetic drift refers to just chance events occurring within populations that cause different individuals to survive, right? Natural selection was really focused, in that case that we talked about, the birds really liked one type of beetle. Genetic drift is really focused on not any sort of directed change, but really just chance event change. And so here you can see a population that has a few green and a few brown beetles. And in this case, somebody happens to step within this population because they happen to step on a couple of green beetles that after this chance event of who they just, happened to step on, it's not that they were trying to hit the green beetles, but afterwards there's a change in the frequency of the different genes in the population where there's fewer genes for the green and there's more genes for the brown alleles in this population. So this is genetic drift. And then lastly, what we have is what's referred to as gene flow. So gene flow is another term for gene flow is migration, but it's not just like a bird flying south for the winter and then flying back up north, because in gene flow, what you need to do is you need to have an individual move from one population to the other population and then interbreed within that population. It's that process of interbreeding within that population that causes the genes from the brown population to enter into this population to create the gene flow. And so when we look at these 4 primary processes that cause allele frequency change within populations, there are 2 that sort of tend to stick out as sort of the major players in what we expect to find occurring in typical natural populations. And that's genetic drift and gene flow. And the reason why is because, well, mutation is relatively slow process. There's not lots of mutation that is accumulated in over short periods of time. And so we typically don't worry about mutation. And natural selection is typically focused on specific characteristics of the genome. And because we're typically interested in looking at the whole genome or the organisms as a whole, that we're typically less interested in natural selection. Now, what's important between genetic drift and gene flow is when it comes to conservation is that genetic drift typically removes variation from a population. And so you can see that here we have a population that has both green and brown beetles, that after our genetic drift event, that there happens to be fewer green. And if somebody else happens to step and maybe hit up, this side of this population, then you would have just 100% brown beetles. So we'd go from a diverse population of green and brown to a population that's less diverse with maybe even 100% brown. And gene flow is the opposite of that. You can see here we start with a population that's 100% green and a different population that's 100% brown, and that gene flow creates diversity, that gene flow introduces diversity into populations. So when we're looking at small endangered populations of natural species, then what we're typically interested in understanding is whether or not genetic drift is going to cause the loss of diversity at a faster rate than gene flow might introduce genetic diversity into those populations. And the underlying premise is that genetic diversity is good for a population to be able to persist because that genetic diversity enables organisms to be able to evolve in some sort of changing environment. And so that leads me to the study that I'm going to focus on today, which is this case study using the striped newt. And the fundamental question here is to investigate genetic structure, which is how that genetic diversity is partitioned among different populations of striped newts to be able to better make conservation recommendations for the striped newt. At the end, when I get to my acknowledgements, I need to acknowledge the U.S. Fish and Wildlife Service and Florida Wildlife Conservation Commission for funding this research. And of course, they're funding the research because these guys have really dropped off in numbers a lot, greatly over the past 50 years, and they're really relatively hard to come by nowadays. So the striped newt, if we look at a little bit of natural history, Their scientific name is Notophthalmus perstriatus. They are a salamander, which makes them an amphibian. So remember, salamanders and frogs make up most of the amphibians. They're small brownish red with a stripe that runs along the sides. They're endemic to northern Florida and southern Georgia. And they inhabit these dry uplands like sandhills and upland pine scrubby habitats. And although this is where they live when they're not breeding, they breed exclusively in isolated wetlands that lack predatory fish because the fish eat them, their offspring, the eggs. And so these are typically depression marshes or cypress domes, cypress ponds. And they're known to have a complex history, which is probably one of the causes of why they're not doing well. They by a complex history, they need these ponds to be able to breed, and they need the dry upland habitats in order to survive when they're not breeding. And so because it requires both of these types of habitats, you need to have good high quality xeric uplands that are located close to cypress ponds in order for the newts to be able to persist. Now, historically, they had this kind of crazy range. And I say it's kind of crazy because there's actually sort of a corridor that runs down the middle where even historically they were not found. And but this is the grayed out areas in the east and the west make up the historic range where they were found. And they've declined greatly through much of this range. And in fact, there are only about 4 populations left in the entire western part of the species range. These are the 4 populations that I've collected samples from. And so you'll see where these are located. And in the eastern part of the species range, they're doing a little bit better. In the eastern part of the species range, they're found in what we call 3 stronghold regions. These are regions of relatively good habitat that's largely connected. And it's in about this region of Florida, from Jennings State Forest all the way down to the Ocala National Forest is this range of these 3 stronghold regions. And then there are a few other isolated populations that we've been able to find and collect samples from. And this down here just talks about listings that globally they're listed as near threatened. In the feds, they're considered a candidate species for the Endangered Species Act. And although in Florida they're not protected, largely because the strongholds occur within Florida, in Georgia they are considered a threatened species. So the research objectives here were to estimate, I try to bold each of these main objectives here, but we want to estimate population differentiation, which is really saying how different is, say, this population from this population or this population from this population. And we want to create a test for what we call a pattern of isolation by distance. And so a pattern of isolation by distance means that we would expect populations that are close together to be genetically similar because there should be gene flow that connects those individuals. But when we get further apart, and then even further apart, we expect populations to be more differentiated from each other. And so when you get populations that are genetically similar when they're close together and genetically more different when they're far apart, that's called a pattern of isolation by distance. We also want to estimate what we call the number of population clusters. And so here a population cluster is a group of populations that we might expect regularly share migrants, and so therefore are more closely, more similar genetically than populations where there might be, much less likely for migrants to be shared. And so really these 2 objectives, the first one and the second one, are relatively similar, trying to look at connectivity among these populations. The third objective is looking at the level of diversity that is present within each of these populations. And so here what we want to do is we want to look at within population diversity, and we want to test whether or not there are differences between those populations that are more isolated. So really, right, these regions right in here are our stronghold, actually including this one here, are our stronghold populations. And all of the other populations are really these isolated populations. And we wanted to determine whether or not there's differences in levels of genetic diversity within populations between those that are in the isolated versus non-isolated populations. To do this, we sampled from 15 sites. 11 in Florida, 4 of them in Georgia. We collected tissue samples. We didn't actually like kill animals to do this. For this type of work that we're using, we can just take a small piece of the tail off of a newt. They actually regenerate the tissue that's taken off of their skin. And so we collected tissue samples from 172 individuals. And then we were able to take this tissue, extract DNA out of the tissue, and then we used a process called RADseq or dd-RADseq specifically. And from this, we were able to find 12,000 or almost 13,000 different loci that are spread throughout the whole genome. And these are just genetic markers that we can use to be able to say, well, what is the genetic diversity at one locus? And then we can compare that across all 12,000 loci to be able to give us a better look overall at the level of genetic diversity for the whole organism and the population or in the region. And so now I'm going to get into some of the results that we've acquired. And so it turns out that if we look at the greatest level of differentiation, the greatest level of genetic differentiation that we see, that this is measured by a statistic called FST, is between this population up here in Ohupi Dunes versus this population down here in Triple N Ranch. And as you might expect, these populations are really far apart. When we look at the smallest estimate of differentiation, you can see it's an order of magnitude smaller, that it's between two populations in the Ordway-Swisher biological region. And as you might expect, well, these populations are really close together and they're genetically similar, and these populations are really far apart and they're genetically different. And if we look at the same level of differentiation across all these populations, across all the sort of the pairwise populations, what we see is that as geographic distance gets larger, so does genetic distance. And this is significant when using statistics. And it tells us that we do indeed get this pattern of isolation by distance when we look across the whole range for these newts. When we use a computer program called Structure to try to tell us which populations are most similar to each other, we get 7 genetic population clusters. So down here are each of the different names of each of the different populations that we've clustered. And then the colors represent the groupings. And then I've made colors on the map so that makes it a little bit easier to see. But for instance, the Apalachicola National Forest and this Dixie Plantation population are going to be both red because they form a single genetic cluster. And what's most interesting from this work is that the stronghold populations, at least these from Jennings and these from Ordway, form a single genetic cluster, suggests lots of gene flow between these good populations. Whereas the other more isolated populations, although they might cluster with a population that's close to them, they're really genetically different from all the other clusters that we see. And then lastly, when we wanted to look at whether or not there was differences in genetic diversity within populations between those that are in the stronghold populations versus the more isolated populations, we see that the stronghold populations, this is a measure of genetic diversity. In this case, it's heterozygosity, which just says, again, how diverse the populations are. We can see that the stronghold populations show a higher level of heterozygosity, a higher level of genetic diversity than what you see in those populations that are not strongholds. And again, statistically, these are significantly different from each other, suggesting that those that are isolated have reduced levels of diversity. So in summary, right, our patterns of differentiation provide strong evidence for gene flow between populations in close proximity. The data are consistent with previous data that suggests that there's no sort of shared genetic clusters between Eastern and Western populations, and that overall we see a pattern of isolation by distance. And then lastly, pairwise comparisons of genetic diversity show that all these all these isolated populations have less genetic diversity than those populations within the stronghold regions. This is important for management. The study confirms that these isolated sites that contain striped newts tend to exhibit very little gene flow with other sites and also exhibit low levels of genetic diversity, right? The problem here is that if one of these populations goes extinct, it's not likely to be refounded by other newts that just sort of wander over there and then resettle into what might otherwise be pretty good habitat. In contrast to these isolated sites, non-isolated populations do exhibit evidence of gene flow within the region and at least between Jennings State Forest and Ordway-Swisher Biological Station sites between these two different regions. And so because these newts are disappearing largely in the western part of the species range, there has been talk by U.S. Fish and Wildlife Service and FWC about doing translocations, about taking newts and moving them from one population into another. And this study would suggest that these repatriation or these translocations ought to occur with samples collected in relatively close proximity to the location that they want to repatriate, and that probably eastern versus western regions should be managed separately if they can. And finally, for us today, this was a good case study to show how we can use genetic diversity to get a better understanding of the natural history and the conservation of a species of Florida's wildlife. A lot of this work was done with graduate students and collaborators. And again, it was funded by U.S. Fish and Wildlife Service and FWC. And with that, I'm happy to take any questions that people have from anything that we've talked about today. Okay, so I can see the questions that are popping up in the chat. And so question 1 says, why do we allow invasive and ecological species to be brought into Florida and sold to the general public? So this is a great question. I think that the majority of invasive species that are found in Florida has lots of invasive species. I have a whole research program where I study marine invasive species and as well as some terrestrial invasive species. And I think the majority of them are not necessarily organisms that are bought and sold within the pet trade and sort of brought in on purpose. But a lot of invasive species either got here through sort of hitchhiking on other vehicles or boats or travel from individuals, so they didn't sort of purposely bring them here. And then the other side of it, I think, is a lot of species that are invasive are from the pet trade and considered relatively benign. So you've got like your goldfish, and then you're moving. And so you don't want to just kill your goldfish, but you can't move it. So you take it to the retention pond behind your house and you just dump it in the pond because that seems better than killing it. Well, it turns out when you do that for different species, that some of them actually can thrive in those ponds, grow to be much bigger than they would ever get in your aquaria and create problems. And so, I do think that the government is working hard to try to limit, lizards and snakes and some of the big predators that can be introduced into Florida. But I think a lot of it is just accidental release, and that's going to be hard to stop.
Okay, Dr. Hoffman, could you comment on the invasion of South Florida with all these boa constrictors and what it's doing to the native population?
Yeah, I can. In particular, I myself did a study on Florida's marsh rabbits, and it's much harder to find marsh rabbits than it ever used to be, because if you go into the Everglades and you find various invasive snakes, that they sort of look like a pearl necklace with all the humps going down them of all the rabbits that they like to eat. And so, I mean, it is a problem, right? And so people will get some sort of small pet snake in their cage and they'll feed it little mice, and then it gets bigger and bigger and bigger. And then again, once it gets so big, then they feel bad about killing it because it's their pet, but they can't really keep it. And so they tend to release it. The thing with Florida is that we have such a wonderful climate that all of these species that get released can survive. And if enough pets get released, then they can actually breed in the wild. And that's what we have going on with these various snakes and lizards, like, Nile monitors and whatnot, is there were enough of them that were released that they can now breed and sustain their own populations.
Okay, thank you, Dr. Hoffman. Do we have any more questions for Dr. Hoffman?
Let's see.
We have two from the
I have one here in the room, Dr. Hoffman.
Okay.
I guess my question is just, what can we do as individuals to help maintain healthy levels of diversity? Like, what, I mean, there's obviously folks like you out there studying it, trying to quantify this challenge, but what can we?
What can individuals do to help foster the diversity and that in wildlife of which you speak?
Well, that's a really great question, and there's at the different level, there's different things you can do. You can buy license plates, like Save Our Sea Turtles license plates, because that money gets from buying the specialty plates gets refunded back to researchers and actual infrastructure projects. But the other thing that you can do is, I think the number one thing that people can do is to be aware of the impacts that we make on the environment that we live. And so I think it all comes from the idea of sort of these classic tropes that we've heard before, but we want to try to use less, recycle, save our environment, don't pollute, don't litter, because these are problems that just really wreak havoc on the land that these organisms need to live in, and there's just not a lot of it left. There was Amendment 1 that was passed a while ago in Florida that was using state money to buy back lands to try to preserve lands. And I think stuff like this is really good to try to preserve the remaining good habitat that we have.
Okay, thank you, Dr. Hoffman. I think we've used up our allotted time, so we appreciate your